it's running.
Thank you. Great. Made all the difference. So today we're going to talk about the comparison of structures of 3D objects. Domenico, I've shown you that slide before, does the video lectures. And in fact, the reason why the slides for the first lecture are still not there is my mistake. Let's fill this now with a few board points that are important. May 12th, there's student assembly, therefore no lecture. 14, Ascension Day, no lecture. 26, Whitsun holiday, no lecture. And Corpus Christi on June 4th, no lecture. The last lecture will be July 7th, and the exam will be in this room at this time, July 9th. That at least is the plan. Provided we have enough space, possibly we have to move it to a different room, and possibly we need another time and date, but this is the current assumption. Makeup is missing here. Should be October 15th, I believe. I'll get back to you with more details on that one. Science is communication, and communication very often starts with questions. Please ask more of those. Here are a couple of questions that I faced recently. First, and that pertains to today's lectures. Why do we compare 3D structures? Why not function? So I'm going to talk today about how we can compare proteins as structures as three-dimensional objects. Why do I do that at all? What we actually arguably want most is really to see that proteins are related by what they do. Why do I compare structures then?
Because structure is feasible. You can cube structure.
So let's just go the other way around. So there's the dogma. The dogma, part of that dogma has to do with structure determines function. And yes, structure is related to function, but it's also we do because we can. It's true. But it's also true that we can, and by this fact that structure relates to function, similar structure relates or tells us something about function. Now, what else could we do? We could, for instance, function. What you think about, for instance, what you could possibly think about is a binding site. We could possibly think about just going straight to the binding site. I call dehydrogenase, ADH5 here. One particular binding site, and simply look up two binding sites. Instead of aligning three-dimensional objects, just align the binding sites. Okay, sorry, slightly more complicated. There could be another binding site here. There could be two binding sites. One is sort of the active, the reason why the protein, the enzymatic activity. The other one could be a sugar binding site that turns this on or off. Okay, then we would have to look at two binding sites. But as you see already, there's a homodimer, the ADH. Homodimer functions as a homodimer, meaning that these two binding sites alone, or these binding sites alone, will not be enough. You also need the protein binding site. Okay, so now we're not going to compare three-dimensional objects, we're going to compare lots of binding sites. And actually we're back to the same story, right? In some sense, we are back to comparing structures at the first place. What's the alternative? Well, the alternative is could look at something on the much more coarse-grained level. They are enzymes. So all that I know about the ADH5 is not how it digests alcohol, but it is an enzyme. On the highest level of some, you may have heard of gene ontology, of some Go tree, this is called an enzyme, and that is one characterization of its function. Why not just compare all enzymes from now on? And there the answer ultimately goes to numbers. We will, over the course of the next few lectures here, get back to why that is so and what this number exactly is. But you can roughly infer 3D structures for, say, half of the proteins. You can roughly infer a function on the level of it's an enzyme for half of the proteins. So if we combine just these two comparisons, you would in fact only be left with 1/4. Throw away 75%. While starting with the structure, all you need to throw away is half. That's another argument. How do we get to shapes? And again, we will get into more detail. Essentially, most shapes, the most accurate way to get shapes is by experiment. Most inferences of shape come by computational biology, and that is what this lecture is partly about. I will talk about it in more detail. So, structure comparison. We talked about the background, and next week we will talk about alignments. So somehow let's begin with more about domains. The alignment, what I mean again by these lines here, protein A begins here, ends here, protein B begins here, ends here. The overlap implies that over the course of this overlap, these two proteins have something similar. And in the case like that, you immediately suspect that, in fact, you have here a common domain between the two. How many proteins have more than one domain?
Most of them?
As in 50-60% or 95%? At least 51, yes, that is true. Does anybody go below or above? So who is for more than 51? Wow. You guys should read the literature more. Why do you say that? Most proteins, according to the literature, don't. So the trouble is you're right. Your prediction is absolutely right on, and the next slide is going to show you. But it's not what you find in writing. So who told you that?
Because we studied more prediction methods, actually not, and those are usually designed for more complex things that you can do with experiments, which I assume is more in the literature. So we actually work more often with proteins that have more domains than that.
Well, so it is true. Let's get back to the word domain. A lot of the data that you look at go like numbers, will match to regions. You have motifs, binding motifs. That's not quite a domain. So this view of a domain as a separately folding unit is a slightly different view. But okay, here are the numbers. Proteins with 1, 2, 3, and so forth. Green, known structures. Blue, not known structures. And we get to why this blue, how we did the blue in a minute. But you see the reason for the literature. If you looked, so the best way to define domains, delineate domains, see where the domain is, you look at the three-dimensional object. For all the three-dimensional objects that we know in green here, not the vast majority, but more than 51%, in fact have only one domain. Yeah?
Yeah. Only enzyme proteins or overall proteins? So
this is the entire data set of proteins for which we know the 3D structure, which in fact is biased by enzymes. Well, we're not entirely sure. It appears to be biased by enzymes. More than half of the proteins for which we know the structure are enzymes, and the current assumption is that maybe more in the ballpark of a third of the proteins, all possible proteins have enzymatic activity. So that would be a slight overrepresentation in the known structures. Somehow answers your question, but not completely, because we actually don't know all the enzymes. But that's the situation. This is the entire PDB.
And can we expect enzymes to have more domains than like non-enzyme proteins?
Why would that be the case?
Why would it not be the case?
Oh.
To assume that like enzymes
so let's
fold.
So let's ask let's ask the first question. How would you answer that question? What's the simplest way of answering that question? That you essentially, do you know of a way that you could do, if I gave you that in the exercises, you could complete today?
What's the question?
The question is it true that enzymes have more domains? The first piece of evidence is not quite answering that question, but it's providing some estimate that could falsify that assumption. Okay, let me get to what I'm thinking. The simplest way is I get a data set of enzymes, annotated as enzymes from SwissProt. I take SwissProt. SwissProt is a database of about 400,000 proteins that have annotations about them. So there you can take enzymes versus non-enzymes. In fact, most in SwissProt are enzymes, so that is yet a higher bias than in the PDB for enzymes. And then I simply look at the length. Okay? If the length distribution would be such that enzymes are much longer, then that would be an indication for an answer. If it would be much shorter, it would be another indication. If they are the same, we unfortunately have to get a little bit more into the detail, because then we have to ask, okay, the length is roughly the same. Does it actually mean that they have the same number of domains? That will make it a little bit more complicated. But there are there are at least two possible outcomes that would answer that question right before we get into the problem of domain. And there is an indication, so there's some data that we have collected at some point that suggests that the average length of a domain in an enzyme is higher, that enzymes are more often than other proteins that have several other domains, signaling domains, attached to them as well. There's a tendency for your assumption to be right because of two ways of looking at it, yes. Back to that question. That was to be the bias in the wrong direction. Okay? So that is green, a data set for which I can safely assume that I can find the domain. Blue is a data set where, in fact, I'm not so safe anymore. And again, I get to that in a minute. But that would suggest the view that you all appear to share, which is that, in fact, even more than 50%, that in the ballpark of less than 30% of the proteins have a single domain, everything else has more. Okay? And that is just here, the difference here. Now, what this slide shows is the number of, it's called fragments here. On the previous slide I called it domains. And here the length. For three different data sets. One data set is PDB, proteins for which I know the 3D structure. One is eukaryotes only in blue. And one is dominated by prokaryotes, or by bacterial proteins. By prokaryotes in this particular case, but in fact has some mix in from eukaryotes. In this particular case has yeast. And there are some historical reasons why we didn't split these two. So let's begin with a couple of observations. The first observation, they're linear. That, you may say, is expected, but does not need to be the case, right? The second observation is that they in fact have a similar slope, all three of them. No matter what the bias with respect to single or not single domains in PDB may be. The blue and red here add up to what I showed previously on the previous slide as blue. The green is still the green. And you see that although the domain distribution is very different, the slope is very similar. That in itself is also surprising. Again, from the data, you have no reason to expect either way. So in that sense, it's not surprising, but it's a statement that is non-trivial. The next non-trivial thing is, when you look at eukaryotes and prokaryotes, you see a tiny difference. They start at a different starting point, and the same is actually true for the PDB. Now, why is that important? So the starting point is not 0, it's 1. Because there's this assumption that eukaryotic proteins and prokaryotic proteins, we went into that last week, have different lengths. Again, would not be supported, also not on the level of domain. The domains look actually very similar. But now comes the, let's call it surprising part. The slope here is roughly a 100. Meaning that the vast majority of domains is about 100 residues long. But not the first one. Okay? So proteins with one domain have on average longer domains, and that is exactly where your enzymes come in the game. Enzymatic domains are slightly longer, and they in fact dominate exactly this difference. Okay, so that is the domain view. Apparently most proteins have multiple domains, and apparently you expected that. Okay, how can we in fact chop proteins into domains? How can we guess them from sequence alone? And what I'm showing you here is a hack. And that hack is the simplest thing you can do. And that is the kind of thing, I'm going to show you something that as a smart student I would assume you can do in less than 3 months. So that is doable easily in a master thesis. The project unfortunately took a lot of funding, more than one person, and more than 5 years. And essentially the outcome is what I'm showing you now. It's very simple, but the devil lies in the detail. And the detail I'm not going to talk about. And the detail killed us in this story. But essentially you do the following project. You have this sequence here. And you now want to parse that into domains. So the first thing, the best knowledge of domains is when we know the three-dimensional object. So we align it to PDB, to proteins for which we know the three-dimensional structure, for which we from the structure can infer the domains. That's the green part I showed you before. Say it's like this. So I find two proteins here in my way of displaying that, for which I can align the domain. I take the longest and I cut out, and I say that is domain 1. Then I have a leftover piece here that is maybe 10 residues long or 15 residues long, clearly not a domain, so I throw that away. I cannot do anything with it. So that may be part of that domain, it may be something else. It may be a functional unit, and that is again back to what you said in the background. A lot of the domains that could be a start of being exported. Signal peptide, so-called signal peptide. They could be 20 residues long. Many of them are in the ballpark of 20 residues long. They are actually cleaved upon secretion of the protein. Many of them are cleaved upon secretion of the protein. So, meaning that they, in terms of a structural comparison, do not have an actual reality. They have a temporal reality. They exist when the protein comes out of the ribosome. They are needed to transport the protein to the cell wall, to excrete it, to get it out of the cell wall, and some of them are then cleaved. Then the rightful assignment of the domain would be like that. Okay, what's the next best thing? What do you know? Once I have this domain assignment, this garbage assignment, and this one I don't know assignment, what can I do now best as a next step to go toward domains? What's the best thing out there if you don't have structures that looks like a domain and that experts have looked at? Anything that comes to your mind? It was briefly mentioned on Tuesday as a data resource. You may have encountered it before. No? PFAM? The protein family database, currently hosted at the EBI since 2 years. Alex Bateman's made it to what it is today. So say you find PFAM regions, and again I showed you the reasons why PFAM domains are longer than structural domains, and I explained to you that actually they are really shorter. This has different reasons why the plot was the way it was. But say we found two PFAM regions, we called them the next best thing to knowing the domain. If I don't know a 3D structure, which I don't at this point anymore, I cut again. So the next best thing that I can then do is cut again, so we would have one domain, one trash item, another domain, and two leftover regions. So the next best thing is to do this kind of evolutionary analysis that I showed in previous slides. So I have some proteins here. I don't know whether you can see the light blue. So there's something here, there's something here, and then you find a protein that essentially has both regions. Find something like that, you cut. I write SwissProt. SwissProt has 400,000 proteins. UniProt has 55 million at this point. Why do you write SwissProt and not UniProt?
Because they're annotated.
Right, they're annotated, but I'm not using any annotation here. Right? Okay, well, again, I'm going to do this for 500,000 proteins. We're not going to look up annotations. At some point, when you care to work on one particular protein, you want the annotations back. And that could be a reason. But in this particular case, annotation is not... The fact that SwissProt is annotated is at this point not why I do that. Can you think about another reason? Or let me put it differently. At this point, I believe if the other reason wouldn't be there, I would rather have the 55 million of UniProt than the 400,000 of SwissProt. Do you know anything? Again, I said SwissProt is annotated. But is there anything else that comes to your mind that may be a difference between knowing a protein sequence and depositing it in UniProt and having it in SwissProt?
Well, the expected quality is higher.
What? Quality of what? That is true. Error of what? Sequences. That is very true. But here's another important point. When you take a genome, particularly when you take human, from the gene to come to the protein is highly non-trivial. SwissProt has annotations. You can only have annotation because somebody did an experiment. Somebody did an experiment means somebody had actually not only that sequence, they actually had the construct, the gene product, the protein, in their lab. So those are actually proteins. While UniProt may be incorrect assignments, maybe fragments, maybe pieces, maybe pseudogenes, or whatever you call that thing. The moment you have an incorrect assignment, you see that this method really gets complicated. Because the method lives off the fact that the reason why you have some here, and some both, is because you have independently folding structures or constructs. The minute that I throw something into my database that has just 20 or 30 residues, or incorrect assignments, incorrect splicings of introns, you immediately kill the entire philosophy of that method. Right? Just a few one could dirten it. Simply because whatever is the signal will be poisoned by something that is an incompletely spliced intron. Right? Few introns could completely destroy many trees. Few incorrect introns. That's the reason here, ultimately. Now, once you do this particular assumption here, that works. So you can cut here, and in this particular case, in fact, we would know that we have 5 different domains. We may argue that this one here still is too long. Maybe one day I'm going to find another, this type or that type or whatever type, to cut it further. But then today, this would be my best assignment for domains. If I now did this, pulled this through, and that is 10 years old now, the work, we haven't redone that. The next question is, how much can we map which part? So the first question here, the fragments that I get at the end of the day, here, in this particular case 5, about 20% of those came from the first step. They map to structures. About 27%, roughly a little bit more, came from PFAM, and very few came from that. This is because SwissProt is small. This is because, in fact, that philosophy has been used exactly to build PFAM. So most of the power of this method is already in the light blue here. That's the major reason why this here is relatively small. And that here is, in fact, the percentage of leftovers. 15%, this one here, and the colors break down, 15% is simply single proteins that are 100 residues long, or 80 residues long, where I essentially know this is a single domain. So I don't need to parse it in any of these. That essentially tells you that at least this part of the pie is a relatively good approximation to domains, and that brings us back, I'm sorry, I should have put the slide back in here, that brings us back to these curves here. So that is why the blue is a relatively good estimate of what we believe domains are. The pie below the one that I just explained here, this one, same colors, slightly different. This is on the level of per residue, and this is at the level of the fragments, slightly different, because in fact these are relatively short, the leftover fragments. So the difference here somehow indicates that what is left over is highly enriched in single domains. So making this estimate that I had before even in some sense better. Okay, let's get back to annotations. 3D, 2D, 1D. And the first one is trivial. 3D is a three-dimensional structure. Okay? And three-dimensional is because you have the coordinates of every single atom in a three-dimensional grid. Okay? What is 2D? 2D structure. Torsion angles.
Yeah.
So I have to think. So my immediate reaction to what you say, I'm not entirely sure. I don't believe so. I don't believe. So torsion angles are local connections. Yes. Locally, yes, but I'm not entirely sure. So there's indications that, can I put it off?
Yeah.
I'm looking for a different answer.
Unfold. Unfold?
Unfold?
Protein not folded.
Is it 2D protein? That's an interesting idea, because you essentially, I like your logic, because essentially what you have is something that could as well condense, because it doesn't have a unique structure anyway. Is that your logic?
Kinda.
Yeah. Is impeccable. So you're right. This is, again, it's not what I was hunting for, but it's an impeccable logic. No, I really mean, if you think about a structure, you understand what it means to think about a 3D structure. We all know that. It's three-dimensional objects. If you think about it as a 2D structure, the way you say is, okay, let's have the freezer in a slide, and that's what most biologists actually do. They put things under glass, 2 slices of glass, which essentially makes them become 2D, and I completely appreciate your point. But I really mean in a less intrusive, in some sense less intrusive for the object. To put yourself onto the level of the atom, where a protein is a big thing, and now your slice technology to compress that into 2D with glass plates is radical. As an atom. So what I'm proposing here is just information content. Okay, let's look at the computer part of your computational biology studies. And then information content. What is a 2D representation of can you think about a 2D? So your angle may go in that direction. For me, the reason why I'm hesitating about the answer is that you can also write angles as a 1D string. And I'm not entirely, that's one part of it. Okay. Too much words. Contact or distance maps. In the distance map, I take the same protein here, I write residue 1 to 90 something, 200, and do it again. And for every single position, I ask this particular residue, how far is residue 1 from all the other 99? Okay? That's a distance map. Now, in fact, I take the 3D coordinates, project it. I could do that with the angles, but in this particular case I project it into 2D, a simple matrix. Now my question to you is, if I gave you that matrix, nothing else, only that matrix, at any resolution that you want, as in detail of the gray zones here. Here what you see is a bunch of black, darkish dots and white, so assume really I had levels in between, which in fact underneath you just can't see it. There is gray on this graph here, and there's even a scalar for the gray. But again, so you had a complete distance map for on the level of residues or atom, whatever you want, for the sake of the argument on the level of atoms. Could I reproduce a structure from that distance map? So in that sense, my question ultimately becomes, and that is where I'm going to get to answering your question, in terms of the information that I need to describe the three-dimensional object, how much did I lose? There was a point that I suppressed. Who said that? You?
So if you basically want to reconstruct a structure from just the distances between atoms.
Or residues.
Or residues.
Let's make it simpler. Residues. Residue distances.
You could reconstruct a structure.
So now, so let's redefine my goal. My goal is that I reconstruct something that if I show it to you on a screen in 3D, you will recognize it to be the same, essentially. Let's not talk about minor problems, and that ultimately is the issue between residues and between number of gray levels between atoms. And the question really is, can I in principle do that?
I don't think so, because we only have the distance, but not the direction.
Okay. Who is with him? Who will tell me how you can do it? If you can't, you're with him. Or think about, not really the details of how you can do, but can you think about any reason how you could, or any process by which you could do it? If not, you should be with him.
I would argue it is too expensive in computing time.
That's not the question. Can you do it? Is there anything that you have heard of that would help you to answer that question? It's not trivial, that's not what I'm talking about. Is it possible, is the question.
You can identify some things like if you do a classification or a linear regression, possibly that you can group them together into
You see antiparallel strands, you see helices, you see secondary structure, and that is now, in some sense, some of the structure you immediately see on these distance maps reveal local features and are similar to angles, are beyond angles, but really the question is, can you get the whole thing? He says no. And I don't hear, okay, let's get back to how we determine structures. X-ray crystallography is one way in which we do structures. I'll show you, I will not really go into how X-ray crystallography does it, we'll just show you some numbers. But can you think about electron microscopy? Can do structures, but they can do structures on the level of blurb, so you don't see atoms. Can you think about any experimental technology that gets structures on an atomic resolution level other than X-ray crystallography? Have you heard of anything else?
other than
X-ray crystallography.
electron microscopes?
No, that is on the. So that's exactly what I mean. So in EM, what you would get, you would not get the arrow. What you would get is possibly what you would get is you would get a large blurb, a large ball, and possibly you would have some dent. So you would see that the surface has a little bit of a hole here, that the overall surface is not actually a ball, but a sort of elongated object. That is the kind of thing you would see. But you would not see the atom. Just that, since we're on it, X-ray crystallography, you take the protein, you do get a lot of it, you actually grow a crystal, and you shoot X-ray beams at the crystal, and it's the same procedure that you see when you go to the subway. The way the images that you see down there at the subway in Garching of the X-ray diffraction. So from the diffraction, you get back to the origin of the atoms. That's the way X-ray crystallography works. But it's another technology that you know. It has to do with magnets. Big magnets. In fact, there are some in the Garching campus. And that is NMR, nuclear magnetic resonance. The way you essentially do, you put them into a magnetic field. In that, you don't need to grow a crystal. You take essentially the raw protein in some solute solvent, put it into the magnet, and the way you excite the atoms is different. What you actually get is correlated excitement. What you actually get is correlations between proximity. What you get is distance. And that is exactly NMR. Since you can use NMR, the experimental data is not that, but essentially it's a distance map. Okay? And NMR proves that since you can get structures from NMR, that already gives it away, right? So you can get from distances two structures. In fact, if you have a complete distance map, you can get the complete right structure. The only problem you have is its mirror image. You cannot distinguish between this distance geometry. It is CPU intensive, but it is relatively straightforward. The only problem, as I said, is you cannot distinguish between the actual image and its mirror image, but that you can do in biology. Okay, you know the orientation of a helix, you know where the protein starts, and this way you can assign, looking at it, you can find which alternative is the right solution. Meaning that you can essentially 100% from the full complete distance map go to a structure. That is, yeah?
We can finish with this. Okay, and is the question, we talked about more flexible elements like loops. Isn't then the moment I take this distance, depending on like, could be the map difference depending on which moment I take this picture?
Yes, and that is in fact exactly what you will see. We will get to that in over the course of the lecture very soon, actually. When you, for instance, here would be the case, that maybe more this long loop here. When you see NMR structures, you actually don't see that. Typically what you will get is a PDB file. PDB has the protein database has the three-dimensional coordinates. And these three-dimensional coordinates you can put into a plotting program and then see this. Now, when you do that for the PDB, you will see immediately when on an NMR structure, there's not only one set of coordinates, there are several. And that is because the NMR model is compatible with different ways of modeling the loop. And NMR spectroscopists argue that is because those are more mobile elements in a protein. So yes, you will get different models, but these different models actually measure or estimate mobility, measure function. And this image is not right. The image that is better is one where the loop has different positions, because that immediately alerts you to mobility, alerts you to its functional aspects. Right? And that is captured by NMR. That is true. Sometimes alternative models reflect a lack of knowledge. So you could also say, I don't know whether it's the loop is here or there because I can't see the detail. Okay? But in some cases that in fact is a strength of NMR, and this is a consequence of NMR. But that is not a problem of the distance map. That is a problem because the distance is not well defined. Sometimes you see that distance and sometimes you see that distance. The moment you take the distance map, you get one structure. So what you talk about is a reality, a possibility that you can see from NMR, but it is not anything that has to do with the transformation of the information from reconstructing the 3D structure from full 2D maps. That has nothing to do with that. Now, again, we take something that is on the level of 3D, boil it down to something that is on the level of 2D, and essentially we do not lose information. That's the important point. Because we, in fact, reduce by one dimension. Absolutely, for all we do. Everything I do in your image is 2D. I show slides, right? I never show you three-dimensional objects. And it's very comforting. What if I go to the next? What if I asked, is there anything such as 1D structure? What would you say? What is a 1D aspects of proteins? String. No, sequence is one. Can you think about another one? So I would, in fact, in my answer to you, I would have argued that angles are that. I'm still not giving you the answer. But can you think about another aspect that you can write as a string? So here are two. Exactly, secondary structure. Here are two. Sequence that is in alignment here. Secondary structure. You simply look at every single residue. Is there an E or is there an H or is there none of the above? And that is what this column shows here. This column shows solvent accessibility. So that simply asks, to which extent is that residue exposed to solvent? So is it something that is more central? Something more on the outside? Now this has some information about proximity, about 2D. But still is strictly is 1D feature. And that now we get around to your comment about angles. You can write angles, backbone angles, essentially at least as two columns. And in some sense they are sort of 1D, since you use two columns, in some sense they are sort of more than 1D. Locally they're clearly more than 1D, but globally I'm not entirely sure. With respect to reconstructing the structure, we cannot fully reconstruct the structure from the set of backbone angles. Not completely. But if, in fact, that is now the problem. If in fact, so in that reconstruction, here the math is relatively simple. For the angles it's not that simple. So if we solve the math, in principle, what I have to think about is whether I could get from angles in principle from here to there. So currently I do that with MD. And MD is just an approximation, while the distant geometry is an absolute, is also an approximation, but a much more analytical, accurate one than MD.
MD?
Molecular dynamics. So it's essentially a random path, a random walk through an energy landscape. Think about it like that as the answer. Anyway, so one thing is clear. Now from 1D, you cannot get back to the structure. You absolutely need, knowing that something is an E. If you know a helix, you, again, locally you have some idea. You know where I+4 will be if you're both in a helix. The hydrogen bond is stabilized by hydrogen, helix is stabilized by hydrogen bonds between I and I+4. So you know, you can put an ideal helix and you will locally know, but then you will not know how to continue. And for a beta strand, you don't know whether here you bind to that one or that one or that one. You have 1, 2, in this particular case is a cutout from there, these two strands. Are they now bound or not? You don't know from 1D. So you may still have an important piece of information, in particular if you combined secondary structure and solvent accessibility, you have a little bit more information and you can possibly piece it together. But absolutely strictly you can't. And you can clearly not go from 1D to 3D. Now, one dimension more and you lose important information. Everything about connections. That is important. In a nutshell, this one we can do by experiments. Essentially, not by computers. This one we can do by computers. This is not that. And that is what the whole field of structure prediction is about. This we can, this we want. We're trying to move this somehow there. Okay, 3D classifications. And that brings us to the issue of comparing structures. Are these two pieces similar? I've shown you before. Now, when I show you the secondary structure, the rods, you begin to see that in fact they are similar. How do we compare objects? Now, let's just look at the level of shapes. You have this circle, you want to see which of these two it fits to. First thing is you simply put it to both places and you ask what is it most similar to. What you implicitly do here is you find a framework by which you can compile the match. And then by this match you in fact have to define corresponding points. So is this now, we are no longer having an ideal circle, so a bent out object here, and we have to now, in fact, in order to distinguish, still see the match, I would see if I had 4 points, then 3 of these 4 points would still match, essentially. So I have to find corresponding points and then superpose. Or in this particular case here, I use 8 points. And then the match can simply be compiled as the difference for every single one of these corresponding points. So I take every point between these two objects and I compile a root mean square deviation. So for every point, I look at the difference of the corresponding point here between one and the other. Does anybody in the room see immediately what the implication of this part of the formula is? Root mean square deviation. And how you could actually write the formula in a simple way, even simpler. So you all have seen this is the way we define vector distances. You have encountered this since school days. And you're very used to the formula. But let's begin with the first question. Can you simplify this? Could you imagine a distance, some distance? Again, it's not the distance you learned in math for vectors. It's not the distance here. Yeah?
Can I take the sum symbol in front of the square?
Oh, no, that you, so let's begin with the factual detail of my question. That would not make it simpler. Yeah, would it for you? Wait a minute. You want to do sum first over the Ri, and then no. You need the difference, right? Or what do you mean? You want to put this?
Yeah, no, it's...
Okay. So no, really simpler. So there's a bunch of mathematical symbols. Get rid of one of them, or change one to another one that is simpler.
You can change the way you measure the distance. So
yes, how?
Go up and.
Oh yeah, that you can. No, stay with the formula here. That's one way of compiling distance. And corresponding points. How can you simplify the distance you compile between 2 points? So yes, you could get rid of the square, but the square essentially is a normalization. That's not what I mean. Okay, assume the square is gone. You cannot get rid of the sum. That's clear. You have to somehow sum over all the objects. Throwing some out of the equation. So you could put yourself on the statement, okay, for most of those I see here that they are similar anyway, can keep them out of the equation. No. We don't know that in general. Okay, that's not my question. But if you forgot about the square root and you forgot about the sum, what can you simplify now? You need 2 objects. You need the I, you need the R, you need the A and B. Okay, guys, what is left is the 2, right? It's not much. So how can you compile a distance? By simply absolute value, right? Instead of squaring it. What's the difference between taking the absolute value and the square value?
We can't get negative distances?
No, absolute value, you don't have negative distances. So absolute value, simply you look at the distance and then you take, if it is negative, you take the sign away of the value. What's the difference between the square and an absolute value and an absolute simple distance? Yeah?
When you take it times 2, it's just bigger, nothing more.
Exactly, but that is not trivial. This means that if you have, so we have 2 situations, say. In one situation, for every point you have a slight mistake. Another situation, in the case of 8 points, 7 match and 1 doesn't match at all. And this will make a bigger difference then, because it's squaring that. So differences, large differences, 1 big difference, and the square counts much more than many small ones. That may be desired or not, but that is an important part of this equation. In vector geometry, you typically are not aware. Variance is the same under the same rule. The way you compile Gaussian distributions. Statistics is part of that. That's an important impact. And in fact, that is the single reason why people shy away from rmsd for structure comparisons sometimes. All the methods that I present and most of the work in the field uses rmsd. But that is the major counter to rmsd. Okay? Something that we never question in statistics or in our simple geometry lessons. Okay, so now we have a measure, we have a way of doing it, we find corresponding points, we superpose, and we compile the rmsd. We have to do some scaling. That is another issue that we have to do. But then essentially we have everything in place that we need to compare these 2 elements to in fact compile simple similarity between these 2 different robots. Now we have this issue but between global and non-global. In a global situation, so you immediately see in your eyes that these are sort of essentially the same symbols, the same robots, everything the same. Now when you look at the global superposition of these 2 by the algorithms that I just showed you, the story is not that simple anymore. And that immediately you see has to do with the fact these elements are shaped because your eye immediately takes it apart into its, let's call it elements. You immediately cut here and you immediately compare the elements and you see that put down to the elements, those 2 objects are exactly the same. Right? Now, the problem, of course, is when you begin cutting, when you see that, in fact, cutting is a virtue, is something that helps you to reveal a similarity, the problem becomes, what is the unit? How far do you cut? In one very extreme case, you cut down to the level of the residue. And that's nonsense. 3D comparison on the residues will give you a sequence, nothing more, nothing less. But also nothing more. So what other thing could we do? We could look at secondary structure. If we did that, we would essentially end up at our alpha or beta, alpha beta, the Michael-Levitt classification, which is very useful, but not as much as we want to do here. So we want to distinguish protein structures on a level much more fine-grained than it has only alpha helices. By the way, for that I don't need 2D, I don't need 3D, I can do that for secondary structure prediction, which will be the first prediction methods that I will. So that brings us to the question, what is the valid unit for the comparison? Well, one way of saying a valid unit is one where the comparison is significant. So, yes, two proteins have helices and I can match the helices. Big thing, most proteins have helices. So there is not a lot of novelty, not a lot of surprise, not a lot of value in that statement. Significant means somehow, scientifically significant, there is a value in that statement. Statistically, how would I assess that? In a machine. Well, again, I could find a structure biologist who can tell me it must be at least 3 secondary structure segments, helix, turn, helix. That's the minimal kind of object that you have to compare to. Say this person, this expertise would not be within reaching proximity to you. How would you go about it? How can you compile statistical significance? Again, the question is, we want to compare 2 3-dimensional objects, and the question really is, how far do you have to cut? And then you will compare those cut pieces. So this is the smallest piece. And that then I will do 3D comparisons on. Think about the robot comparison that I showed you before. So how far do you have to cut? And how could you estimate what is too small or not too small? Well, you need a database of protein structures, of shapes. Say for your comparison, that's the signal. And say that is the signal, the match of that fragment that you looked at here with respect to the database. Then the significance of the signal comes from how far that value is from the average and how far the spread of this distribution is. The further away from the average you are, and the smaller the spread, the more significant you're finding. So in that particular case that I draw here, you would have a fragment that has an absolutely significant similarity. So you would have something that is non-trivial. Again, whether that then implies functional similarity, whether that in fact leads further, is yet to be discussed. But at least you would have in your machine, before discussing with an expert, you would have something that indicates that what you cut as a unit is something that is worthwhile looking at, right? And you're not using anything that you do not have in your computer right here at this moment if I close the door. Without internet. Absolutely trivial. And that then would allow you to know where to stop cutting, and that would allow you to compile this, and then you essentially have done the superposition and everything is fine. Now, you could slightly simplify the finding part by looking at the matrix of differences here. You essentially would compare one matrix, the internal coordinates in some sense, like the angles, but in this 2D, and then you would compare 2D matrices. That is the same problem, but slightly different. Now let's project this to the issue of proteins. And some of these slides come from Patrice Köhl at UC Davis. First, we have to identify equivalent positions. Then we have to find the superposition independent of domain movements. So in this particular case here, we have 2 different fragments that map to 2 different sides of the molecule. Then we do an rmsd and compile rmsd values. Structure biology typically argues in angstrom. 1 angstrom is 10 minus 10 meters, so it's 0.1 nanometers. And in this particular case here, I have 2.8 angstrom rmsd main chain difference. What that means is I take only C alphas between these 2 objects. I find the optimal superposition. The corresponding points are the C alphas in this particular case. For all the C alphas, I compile the root mean square deviation and summed over all the fragments that I align here, that is 2.8. This one here is essentially also 2.8. The point that is slide makes here is that the comparison is not metric. Meaning that from the fact that A is similar to B, and the fact that C is similar to B, you cannot infer that A is similar to C. And that, in fact, the triangular inequality is one of the features of a metric that is not satisfied here. Because the comparison is essentially local. You look at two different things. Now, my question to you, is this a better fit to that than this one to that? So in your image of the glass that you compress a protein to 2D, this is sort of a more compressed sheet than this one, right? While I would argue that the helix here looks very, very, very much like the other helix.
The problem is also length of CO, because what happens, so C is shorter than B?
Mm-hmm. C is shorter than, trivially C is shorter than B, but C is also apparently shorter than A.
Yeah,
that's the more relevant one here.
But my question is for the rmsd, so I only compare what I can compare, where there are atoms that I...
That's the difference between a local and a global alignment, or a global superposition and a local superposition. In this particular case, and we will get back to that issue, but in this particular case what I show you is in fact a local superposition. Yes, I compare only what I can compare. Which does in fact bring up the issue here.
So in that
but
sense,
very,
A is maybe better because it looks like it's low.
It's more, exactly. You could say, and now let me give you another piece of evidence here. What I never showed you, but this is, no, this is not true. Okay, that's a valid point. I wanted to go somewhere else. And actually I don't know the details, you could be right. I never realized that. Usually when I show this slide, and nobody in the room makes that point, and you could absolutely be right. Usually what I want to point out here is that the difference is really the part of the helix that is keened away. So you have a small region for which you have a big difference. And a small region for which you have a big difference is exactly the thing that hits you in an rmsd, in the square of the difference. Now back to how could I compare the entire thing? That's the same problem here. If I wanted to compare this whole thing and whatever goes here, then the region that is different would completely dominate the comparison. The more you're different, the higher you would count for the rmsd. The more things that you actually cannot compare will make everything look like different. Right? That's the problem again with rmsd. Now, here is a method that, in fact, originally from Willie Taylor and Christine Orengo. Willie Taylor shown here, and Christine Orengo. And SAP is one of the first algorithms that compare structures. 89, we will get back to that next week. 86 is when the first alignment method came from Temple Smith. There was another one before that, but this is sort of the beginning of alignment. And that one uses dynamic programming. This is the same algorithm of dynamic programming applied to compare structures. Not as trivial to compare structures with it, because the 2D information is not easily cannot be easily put into dynamic programming, and the brilliance really of SAP is that Willie Taylor found a way to look at a double dynamic programming. Double dynamic programming is essentially you do dynamic programming on distance matrices. So you create local distance matrices of the two proteins A and B that you compare, local vectors, and then you essentially compare the local vectors between two different matrices. And you do that in a formalism that is in fact copying, generalizing dynamic programming to the case of 2D objects. highly complex. That was the first structured alignment. Dynamic programming always needs an objective function. That is the objective function you have for the local vector. And the downside to that is you have some loss of information about how the vector is oriented. But that is one valid way of doing that. There was a refinement put in where you also need use some sequence information, because aligning structures without sequence, Willi argued, is not good enough. You also want to know whether you map hydrophobicity of residues, not only because of the shape, and that you can easily just put the formula down there where you can put weight matrices in here, and you can easily account for that. Here's another way, another solution that comes from Lisa Holm here, now at the University of Helsinki, Chris Sunder, who is at this moment still at Sloan Kettering in New York, called DALI. That is distance alignment. And let's get back to some of the players. Chris Sunder is one shaper and shaker in this field. Chris has that's May 2011, over 770 papers. One cited over 6,000 times, 6 cited over 1,000 times, 87 over 100, H-index of 92. Is a fellow of the International Society for Computational Biology, and that is the event at which he became the Senior Scientist Awardee. So this is extremely high. Have you seen H indices? This is, as far as H indices go, this is stellar. Okay, here's the idea. DALI, distance matrix alignment. You essentially look at these two distance maps and see how two distance maps agree with each other. Since we do not globally align proteins, we're not looking at the entire distance map, but we cut out regions. We look at the similarity between two local fragments. Okay? Now you essentially move around to the objects and you compare differences between two matrices. The optimal path through this is not trivial to find. That's what Monte Carlo is used for here. So you find a random path through the comparison of these two distance maps, and at every cell, essentially, they begin with hexapeptides, 5 mers. At every cell, you simply compile the distance between two distance matrices. Okay? You can do that because 2D is completely compatible to 3D. I mean, you can, from a 2D object, you can come to a 3D object, yet the information on 2D is simpler. All you need to do is entries to a distance map, and you essentially do differences of matrix entries. That's totally trivial, right? The problem here really is to find the optimal path. In the other case, that corresponds to finding the optimal superposition of 3D objects, right? DALI. Voroline comes from Fabian Bürzele and Ralph Zimmer. Many of you would know Ralph. Fabian was a postdoc who studied bioinformatics here in Munich, and that was a method they published in Bioinformatics in 2006 that uses Voronoi environments. So now the idea is, in some senses, again, you seem to like the 1.5 answer to any question I come up with. On the one end you have a 3D structure, on another end you have a 2D structure, and somehow this is in between. It looks at local surface or local environments that define a particular residue in its environment. You could think about it as a plaque, as a click in a distance map. But you can also think about it as like a sphere cut out from a protein, and then you do simply local alignments of regions. Somehow it's in between. Now, the beauty of this is that the Voronoi environment is not defined by me. I'm not saying 5 mers. The Voronoi environment is defined by the structure. So I can change essentially the entity of the comparison according to how the structure looks. And that is one of the great aspects. They then get to the level of comparing matrices again once they start here, and thereby map it to a problem that has been solved, for instance, in DALI or before DALI in other methods. There are many other comparisons, And I'm just going to go to another aspect of this. And that aspect has to do with rigid superposition. So here we have two forms of carbonyl. One is ligand free and one is with a ligand in the center. So they all have their binding sites, but here is another larger molecule binding. It's the same protein. It's the same three-dimensional shape, or not, because in fact upon binding you clamp into the binding. And you change your 3D structure. How can you identify that similarity? Again, only by cutting. Now, cutting gets us into the old problem of local versus global. So what is better? I have an rmsd of 15 angstrom for 143 residues, or I have an rmsd of essentially here the bindings, the calcium binding site, less than an angstrom for 62 residues. That you may immediately argue, and again, so the two structures are green and red here. This clearly is a fit of the binding site that is important, while in this particular fit, you may argue you do not quite see whether that is biologically meaningful. Example where you clearly can answer, oh, local alignment in this particular case tells the story, the global one doesn't. But I will show you examples where that is not so simple anymore. This is just a few of other methods that are out there to compare structures. They do use different features. They use some of the common technology that I prepared, that I showed or briefly introduced. Here's a paper from Rachel Golodny, who's now at the University of Haifa. Patrice Köhler already mentioned, and Michael Levitt already mentioned, Stanford University, in which they simply took a bunch of methods and compared how well they performed to align three-dimensional objects. Well, the first question is, how do you in fact identify what is right? So we take PDB and we want to compare methods that align, structurally align to proteins. What is my standard of truth?
Structure from the PDB?
They are from PDB. So every method that I'm comparing, so I'm going to compare SAP, I showed you one, DALI, those would be two different methods. And I want to say which one is better. Both of those align to structures from the PDB. Well, one, no, I'm not saying anything. How do I find out which one is better?
We need a set of different structures at first.
Yes.
Only helix, only sheet, both, so a mixed set to test on, first of all.
That's a great idea. So let's just get a set where we have, say, blue things that are similar structures and red that are absolutely not similar structures. And if one of the two methods that I compare, say I compare two methods, and if one of them makes no difference between these two data sets, then clearly that one is worse, right? That's your logic. Is that true? Can
you repeat it, please?
So I have, I don't have blue and red, but I have black and red. So not 3D similar as a data set, and that is 3D similar. Those. Now I have method A and method B. And method A says that the value for all in here is, I don't know, let me just invent a number, is 3. In here is 3, and in the other one is 3. And method B says red is 5 and black is 2. Since the black ones are the ones that are similar and the red ones are the ones that are dissimilar, and since method A gives the same result for both, method A obviously cannot distinguish between things that are similar and things that are not. So you're right on. If the outcome would be as extreme as that, you would immediately see something. Now let me derail your idea. So far I'm at the point where everybody would agree that that was a good idea. Let me derail the idea. Essentially what you say is alpha or beta. This here, B, could be a secondary structure prediction method. I would get B right. Because it clearly can distinguish between all alpha and all beta. It has no idea of comparing structures. But it absolutely can do that task. So it would appear to be whatever a structural alignment method will do, it will never be as gross in the difference as these, or most likely. These two values, most likely for any structure comparison, will be closer than for secondary structure predictions, simply because the secondary structure prediction is so simplified that we'll absolutely see no. It's too stupid to see any similarity. And even between alpha and beta, structural alignment will see some similarity. There will maybe see a difference here. So there would be more the situation where method A has say 6 for this and 1 for that, but or 2 for that. But method B has really 50 and 1 or something like that. So method B would appear to make a bigger difference simply because it's too blind. That's the problem of your idea. It's a good start, but it may not only be a good start, it may be a great first level filter as long as we're aware of that. But it's not enough, right? So if we assumed that structural alignment methods sort of essentially align shapes, then essentially for this big difference, they should all somehow do it. Again, we have a good first filter, but how can we refine that? That's very simple. You need a bunch of proteins for which you not only know that they are not similar, but that they are similar. So you simply take yourself your best expert, but you didn't reach in the first question, or the previous question I had, and ask, give me for 100 proteins similar, not similar, and differentially more similar, less similar. We could do that. But could you imagine a way in which you could even sort of help this in terms of getting more numbers? Well, you could, for instance, look back at annotations. So now you look at things that have similar annotations and similar structures according to that expert, or something like that. Now what you cannot do is take 5 different methods and say, I will take everything as black where 5 methods agree. Because then you can no longer distinguish between those 5 methods. And it's in fact a tricky issue. So they took expert classification from SCOP, and I will get back into what that is. Plot a fraction of false positive, fraction of true positive, and see for these alignment. I talked about SUP, I talked about DALI. This is more geometry here. Waterline is not in here because it was before Waterline. Well, not quite, but yes, it was before, 2 years before. Low is not good. You want to be somewhere up here. And the first observation is that, in fact, there's a difference. The second observation is that, well, I didn't tell you that before. So this is if you, for every single method, use the best of that method. So you have 5 different methods, and one of them is the best. You take that. And you do that for all the proteins in the data set, then that's where you would get. So if you had 5 methods, or in this particular case, 5 methods at your disposal, and if you could correctly pick the right solution every single time, you would clearly do better than anything else. Trouble is we can't do that. But in some sense, that up here shows you that the solutions you have in principle contain the right information if you just could get it out. And in fact, some of those here, down here, are the original methods. And up here is Rachel's refinement, or Rachel's refinement to that original by trying to pick better, given the energy, the superposition she sees. She did not really develop a new alignment, but she found a better way to use the scores of the given ones to go up. So, and in fact, in some sense, the fact that this still is higher means there's still room for improvement. The fact that some of these methods are better than others means that you could also possibly still gain from getting better methods.
Question, doesn't that mean it's biased against a certain data set? So
what? Yes, so sorry. That could be the case, but it doesn't mean that. I cannot answer your question. You're right. That is a very fair suspicion. And that would, if you were right, but the data set is biased by a particular method, then that would also explain what we see. Now, the fact is that this data set that they used was done by somebody looking at a structure. And that somebody is called Alexei Murtsin. Now, Murtsin looks at a structure means he uses geometry. Now you could argue geometry is most similar to some of these methods. So you could imagine some bias, but it's not that trivial and it's not that clear and it's actually not what we believe it is. But we cannot I cannot prove to you, by no means can I prove to you that this assumption is not right. And in fact it's rather the opposite. Alexei Murtsin is biased by visual classification, because initially he did that all visually. He pulls in sequence information, so and there are some methods in here. So CE is in fact lower, and CE is less using this sort of visual thing. So I could pull out from their paper evidence for your suspicion, but it still is not what we believe. Okay, I will continue on this one here with a few images on databases. SCOP, the database used here, is one of those aspects, and the way we see protein structure space next Tuesday. Thanks for your attention.